Right, so we are now being recorded. This is our seventh video teleconference for BIOL 105L YC. We haven't met in a video teleconference for almost a month now because of the midterms week and the week after that one. But if we look at our calendar, we are actually now back in sync with the original number of video teleconferences. We are on our seventh. And by this time of the week, you should be finalizing your reports for exercise 8. That's PCR. And the agenda that I am going to have in this video teleconference is actually for us all to review on exercise number 7, and we are going to connect that to exercise 8 PCR. So I want us all to be on the same page and to realize how these three exercises, 7, 8, and 9, are related. Okay, so if you might have noticed, exercise 7 was quite short. You could have done it in 30 minutes if you were the ones doing it in the lab. And exercise 8 also has very simple and short steps, but we had to wait long because of the PCR. But nevertheless, it was quite straightforward, no? And then next week, we are going to see what's gonna happen in agarose gel electrophoresis. So, um, what am I saying when these three? What am I meaning when I say that these three are actually related to each other? Um, First, we did DNA isolation. So we got the material from inside the cell in the form of DNA. And we all saw it was from my cheek cells. So those were my DNA. And what do we do with the DNA? Now, in the actual laboratory setting, when you extract DNA, you would actually do something with that extracted DNA. Meaning to say, it's not the end of the objective to extract the DNA. So that's why in exercise 8, we are doing PCR. And then later on in exercise 9, we will be doing AGE. So without further ado, let us review what we're done in exercise 7 and also exercise 8. So before that, I forgot to ask you, um, may I hear your voices so that I'll know that you're still paying attention? Good afternoon, guys, again.
Hello, sir. Good afternoon, sir.
Afternoon, sir.
Good afternoon, sir.
Good afternoon, sir.
All right, thank you very much. All right. So I hope in this video teleconference, if you were still lost as to why do we need to extract DNA, what is this PCR thing? I hope that after this video teleconference, it will be clear to you why we are doing this. So let's start right away. In DNA isolation from cheek cells, I have sort of used pictures here to outline the steps. Now we have the ones inside the red box and the ones in the green box. Now, can anyone from the group help us recall, okay, what we're done or the steps that were involved here as highlighted in the red box?
Hello, sir.
Yes, please.
In the red box, the first step was the cheek cells were sort of chewed out from the gums, and um, there was it was also introduced with um, NaCl solution. It was gargled and then put in if I remember, a falcon tube, and then uh, I think 20 microliters of it was put into uh, the
Microcentrifuge.
a microcentrifuge tube, and it was centrifuged so that the chick cells will be pelleted out, sir.
All right. So technically, no, the steps that you said were correct. But just a correction, the cheek cells were not from the gums. They're different things, cheek and gum. So at the sides of the mouth, no, the cheek, the inner surface, the cells there are quite easy to slough off. No, so they're like your skin that they are shed every now and then. So a simple gargle with isotonic solution, so we use isotonic salt solution, would actually um, detach some of those cheek cells and mix them in the solution so that when the one who gargled spits out the solution into the falcon tube, there are also cheek cells that come with it. Okay, so that's what you were saying a while ago. And then 1 ml. Okay, we get 1 ml at a time from our falcon tube. We put it in our microcentrifuge tube, and then we centrifuge this, and then we see that the cheek cells are pelleted out, okay, as you mentioned. And then we discard the supernatant, the liquid on top. But then our goal is to have cheek cells pellet that's as big as a matchstick head. So we repeated it 3 times in the video, okay, um, until we got that pellet that's as big, more or less, as a matchstick head. So that pellet contains the cheek cells, and the cheek cells contain the DNA that we are interested in. Okay, so that's for the first part of exercise 7. So let's go to the ones highlighted in the um, green box. So can anyone in the group recall to us what we did in the next steps? So we are now here in this portion of the procedures. We now have our cheek cells. What's the next step? Anyone? What chemical did we add with our cheek cell?
Hello, sir. Uh,
Yes.
the chick cells were then introduced with instagene matrix, and then it was then incubated in this water bath at 60 degrees Celsius for 10 minutes. And then after the first 5 minutes, it was removed, and it was then um, what's it called?
Vortex.
Vortex, and then for the remaining 5 minutes, it was incubated again in the water bath, and then it was then dried off and then put in this dry bath at 100 degrees Celsius for 5 minutes, sir.
All right. So those were correct. Okay. So what do we do with how do we add together the cheek cell and the instagene matrix together? If you can recall, we had a new tube, the screw cap tube. We placed here our instagene matrix. Okay, I'm gonna admit someone first. All right. So we placed our instagene matrix first into this screw cap tube. If I'm correct in recalling, around 1 ml of it. So the instagene matrix is not just liquid. No, if you notice, it actually contains those beads, translucent beads that go to the bottom of the tube. No, so para siyang it's a mixture of water and sand. So parang ganun ang instagene matrix. And then we added all of our 20 microliters of our DNA sample into this instagene matrix. Okay, so that's what we did after that. After getting the cheek cells, and then we incubated, as you mentioned, first at 60 degrees Celsius using water bath method, and then later on at 100 degrees Celsius using the dry bath. So my question is this, since this is already done in your lab reports, what is the function of this instagene matrix? Anyone who can recall from the lab reports that you made, what is the instagene matrix for?
Sa akong na-remember, sir, is that the instagene matrix was used to chelate out metal ions.
You are correct. Okay. So the beads in the instagene matrix will sort of attract and trap the metal ions that are present in the solution and in the cells. Okay, so metal ions. Um, so that's very important. But my question is why? What will it do if it will chelate away the metal ions? What is the effect of that? So you are correct. But what is,
Sir,
yes,
the metal ions serve as cofactors for the DNases which are present um, in the solution from the DNA sample, and what it does, the instagene matrix, is gets rid of the cofactors essentially, and therefore disabling the activity of any residual DNAs from degrading the DNA sample, sir.
yes. So perfect. So correct, no? So the ions that are chelated by the instagene matrix are cofactors of enzymes that will actually degrade or lessen the quality of the DNA, okay, that we will be getting. So because we wanted to preserve this for the next exercise, no, or for the next step, we need to prevent those enzymes from doing their work. How do we do that? We prevent their cofactors. And how do we do that? We use the instagene matrix to trap the cofactors. Okay, so that's the purpose of adding the instagene matrix. Next question, why do we need to incubate or to heat our sample? What will the heating of the sample accomplish? Anyone?
Hitting the sample, sir, allows for the lysis of the cell and release so that the DNA will be released, sir.
All right. So our cells are eukaryotic cells, meaning to say the DNA is stored not just within the cell membrane, but also within the nuclear membrane. So both the nuclear membrane and the cell membrane need to be lysed first, as you mentioned. And in order to do that, heating the sample for a short time will accomplish that lysis. No? And once the lysis happens, the DNA will be released and our instagene matrix is already present. So if the DNA says the enzymes are also released, their cofactors are going to be attracted by the instagene matrix, so they cannot do their work to degrade the DNA. Okay, so that's everything for DNA isolation actually. So after this, we refrigerated our sample for the next step. Okay, so by itself, of course, there is no really reason to this particular exercise. Why do you want to extract DNA? Okay, in reality, when you're doing this in the real lab for a real purpose, it's actually to extract DNA from certain samples. So for example, if you work in forensic analysis, you might need to extract DNA from certain cells like blood cells or even sperm cells, di ba? Example. Or if you want to identify, for example, bacteria that you do not know, you just get some cells of the bacteria and then you extract DNA from the bacteria. Okay, so that's really the purpose of exercise 7, simply to extract DNA. Now, what do we do now with our extracted DNA? Okay, so in this slide, you can see 46 chromosomes, 23 pairs. So these are human chromosomes. Okay, so what we have extracted in exercise 7 is actually the 46 chromosomes of myself. Okay, those are my 46 chromosomes there swimming in those instagene matrix thing. Okay, so which part of my genome, because this is the genome, no, all of the DNA is my genome in the 46 chromosomes. Okay, um, which part of the DNA are we interested in? Okay, because you cannot study the whole DNA, you just want to study the portion, and the answer is it's somewhere in chromosome 16. Okay, now if you read your exercise number 8, which portion of chromosome 16 are we interested in?
The PV92 region, sir.
All right, so there is a region called PV92 of chromosome 16, and that region actually contains a sequence called the ALU sequence. Okay, the ALU sequence is situated within an intron. We haven't discussed DNA yet siguro in genetics, no? Wala pa. That will be next week or tomorrow for those of you who are in YC. The allogene is in an intron, meaning to say it is not expressed or translated into a protein. Okay? So it's a gene, yes, but it is not that important. If I have it, I'm okay. If I don't have it, I'm also okay because it's in an intron anyway, right? Okay, so we are interested from my DNA sample about my chromosome 16 if I have the allogene. What am I talking about? There are three scenarios no, for me. First, I could have both allogenes in both of my chromosome 16. Di ba? Pair yung chromosome, yung homologous pairs. So first scenario here, this first two, I could be positive in both of my chromosomes for the allogene. Second scenario, I could be negative and then negative for the allogene in both of my chromosome 16. And last scenario, I could have one allogene just on one chromosome 16 and the other chromosome 16 is negative for the allogene. So this can actually be translated as my genotype, no? So I can be a homozygous plus plus, I can be a homozygous minus minus, or I can be a heterozygous plus minus for this allogene. Okay, remember just the chromosome 16. The question now is this. When we extracted DNA, we extracted all of the chromosomes. But our objective is just the allogene in chromosome 16. How do we target that particular gene? But you cannot see the DNA. Can you use a pair of scissors and then just snip the portion that has the allogene in chromosome 16? That's impossible. You can't even see DNA. Okay? Let alone cut it. So my question to the group now is, how do we accomplish in zeroing in isolating this chromosome 16 allogene? Anyone? This is our only target. We have the whole DNA. So we have a problem. How do we just get this and not the whole DNA? Can try answering if you have a wild guess, an intelligent guess. What do we do to get what we want here?
We use primers, sir.
Yes, yes, okay. Primers are very important. Okay? But what is the process where primers are used in? It's actually a title of your exercise. What is it?
Polymerase chain reaction, sir.
Yes, okay. So it's PCR, all right? But before I go to that, let me jump to this first. If I am homozygous for ALU, positive, positive, meaning to say if I have ALU in both of my chromosome 16, what we'll get will be a 941 base pairs of DNA. Now, if we try to isolate this. Now, look, if I'm homozygous negative, what we'll get is a 641 base pairs. It's shorter. Why is it shorter if I'm homozygous negative? Can anyone guess why is it shorter? By 300 base pairs, right? 641 plus 300, 941. Why is it shorter by 300 base pairs?
The allo sequencer only has 300 base pairs, sir, theoretically, sir.
All right, so that's correct. So if we're gonna draw here, this portion alone, that alone is 300. So difficult to draw. Okay, there, 300. That's 300 base pairs. So naturally, if I don't have that allogene, then I will be 300 base pair short. No, this one, the homozygous negative. So naging 641 lang. Okay. And if I'm also heterozygous, I will have a mix of the other half will be long, 941 base pairs, And the other half, the one at the bottom here, will be shorter at 641 base pairs. And as one of your classmates mentioned a while ago, we are going to isolate this gene by using polymerase chain reaction. So PCR is actually a way of gene amplification. Okay, specific gene amplification. Meaning to say, there's a specific gene that we want, then we will amplify it. Meaning to say, we are going to replicate it many, many times. Billions of it, in fact. Okay, so that's what we did in the PCR. Okay, and we will look at that now. So before we go to the next slide, are there any clarifications on these illustrations? These were shown in your lab manual, but maybe you had some questions while you were reading it yourself. And maybe you can ask those questions now if you still don't get the answer to those. No clarifications? Then we will move on to the PCR, okay? The PCR. So PCR, or polymerase chain reaction, it's just the artificial way of replicating your DNA. So again, in genetics, we haven't discussed yet DNA and DNA replication. We will do that shortly tomorrow for the YC section. And so PCR is the in vitro version of DNA replication. Now, this one, you can do this in the lab. And you can replicate a DNA segment many, many times. So there's a specific target, in our case, the allogene. And how do you do PCR? You need a cycling between heating, cooling, and then heating a bit again. Heating, cooling, and replication. So in your lab manual, this is what it was called, no? Denaturation. That's the heating at 94 degrees Celsius for 1 minute. Annealing, that's cooling at 60 degrees Celsius for 1 minute. And then extension, that's replication at 72 degrees Celsius for 2 minutes. And then in the video, you saw that we repeated this 40 times in the machine. Okay? And it says here it just takes 2.87 hours. But if you were observant in the video that I posted, it took 4 hours. Now, why was it 4 hours when it's just supposed to be 2.87 hours? Okay, the answer to that is, before the machine proceeds to the next step, it needs to reach first the desired temperature, di ba? So for example, from denaturation from 94, next step, the 94 degrees Celsius temperature of the machine will go down first until it reaches 60, and only then will it start timing for 1 minute. So there is a lag between the steps because we are waiting for the temperature to go down or go up. So that's why we took longer than 2.87 hours. It was 4 point something hours long. Okay, so these are the steps. So be familiar with these. No? So to illustrate it differently, this is the DNA that we got. So my DNA. Okay. And we only wanted to isolate a certain portion. So this blue portion here, and you can see I placed there alu, because that's the allogene. That's our target sequence. Okay. So how do we replicate the allogene alone and leave the other parts of the DNA? All right. So here's the first step, denaturation. So you need to heat your DNA sample at 94 degrees Celsius so that the two complementary strands will separate. They will unzip. Okay, so that's the term denaturation. Okay, so once they have unzipped, you have achieved denaturation. Right. Next step, annealing. Annealing means the sticking, no, or the combining. What is combined or stuck? It's actually these light blue oligonucleotides, the primers, are going to stick. And look where they are sticking. they stick exactly at opposite ends of the allogene, di ba? Okay? No primer sticks at the gray region. It's always in the dark blue region and at the ends. Okay? And that's very important, no? One of your classmates mentioned a while ago, how do we isolate the allogene? He said primers, and he is correct. Because look at this. The primers will only stick to the opposite ends of your allogene and nothing, nowhere else in the DNA. So next, extension. So with the help of enzymes, shown here in yellow, the primer is going to be extended, in this case going to the 3' direction, and also in the 3' direction in this case, until it reaches this portion where the allogene ends. Okay? But notice here, this one continues, so this is not quite our target yet. But later on, we'll see how it's going to be accomplished. Right. So now, you have this allogene that's sort of replicated already. Okay. And the way we cycle between these steps, 40 times, is through the use of a thermal cycler. Okay, so this machine is very expensive for the reason that it can change temperatures at the exact time for the exact number of times. So it's called the thermal cycler. And I don't want you to mistake PCR for the thermal cycler, okay? This machine is not PCR. This machine is called the thermal cycler. It's used to do PCR, okay? Because when I was your age also, I was also confused. I thought that this machine is PCR, but no, it's thermal cycler. The PCR is the process, of replicating the gene of interest. But using the thermal cycler alone is lacking, May kulang pa. Ano ang kulang? Kailangan may mga ganito. Okay, the one here on the left. What are these? Okay, so first, what is this I'm pointing at? Anyone?
The primer, sir.
All right. So there needs to be a primer first, Specifically two types. Why two types? Look at this. There's a primer that sticks in the 5' end in this portion, and there's another one sticking to the complementary strand. So there needs to be two primers, otherwise only one of these will be replicated. So two primers, correct. What about this? The yellow blobs, what are those?
The Taq DNA polymerase, sir.
All right, so the Taq DNA polymerase. Okay, so let's pause for a while. Why is it called Taq polymerase?
It was isolated, sir, from the um, heat-resistant bacteria called Thermos aquaticus. So that's why it's called Taq, sir.
Right, so that's correct. Why cannot we use human DNA polymerase?
It could not, human DNA polymerase could not withstand too high temperatures, sir.
All right, so you notice here during extension, that's at 72 degrees Celsius. Our body temperature maxes at around 37.6 degrees Celsius or so that our enzymes work optimally. So if you put a DNA human polymerase here, it will be denatured. No, it will not work. So correct. So you need Taq polymerase instead. And then what about these dark blue T-shaped things here in the tube? Anyone? What are these? How do you call the monomers of DNA?
The nucleotide, sir.
Alright, so you need nucleotides as well. Di ba? Kasi nagbi-build up itong Taq polymerase ng new strand. Eh. So kailangan ng source of new nucleotides. So it's also included here. And we also have the magnesium 2+. What's the role of this magnesium ion?
The magnesium ions, sir, also act as cofactors, sir, for the Taq polymerase to do its job, sir.
All right, so it helps Taq polymerase do its job, do its work as an enzyme to extend the DNA that you see here. So it's important. Remember, you mentioned a while ago the function of the instagene matrix. It chelates the metal ions. So we should be careful when we put our sample here that we will not include any of the beads from the instagene matrix. Otherwise, our PCR, no, the process of PCR will not work. Okay, and of course, you need here a buffer. So there needs to be a buffer to balance the pH. And you mix your DNA sample. So how do we call this oligonucleotide primers plus Taq polymerase plus nucleotides plus magnesium ions plus buffer? How do you call that mixture?
Master mix, sir.
Okay, so this is our master mix, and we add our DNA sample to this, and then we are ready to run PCR. No? We can then put our sample with the master mix in the thermal cycler and do PCR. Okay, so if we continue now our illustration, we can see here in this upper portion that indeed the allogene are replicated, but there are bits and pieces of gray strands, no? May mga extensions. We don't want that. But if you look at here at the bottom, okay, this one that's highlighted in white, it's completely cut off. And it's just the allogene. And there's another one here that's also just the allogene. So this is what we are interested in. If we do the PCR cycle 40 times, we are going to produce 2 to the power of 40 molecules of the allogene, or 1 times 10 to the power of 12 molecules. So that's a lot. Now, so we are amplifying now the allogene and not the whole DNA. So I hope you got the point of the why we did PCR, because it allows us to focus on just a very small portion of the DNA that we extracted. And as your classmate mentioned a while ago, very important is to decide what primer to use. Now, lucky for us, in the demonstration, the primer there is already pre-prepared or pre-chosen for the method. No? So I don't have to look for a specific primer. But if you are a scientist and you want to isolate a gene, then you need to be aware that you need to engineer your primer first. Okay? Now, what do I mean by engineering your primer? You dictate what sequence your primer will be having. Is it A, C, T, G, or A, G, C, T, etc. Okay, so that's actually an exciting job, no? If you are going to be a scientist and you're going to isolate DNAs, you're going to be more knowledgeable in that area. All right. So, but for now, we settle with a pre-made primer and we are able to do PCR. So in the next exercise, the result of our PCR, we're going to put that through gel electrophoresis, no? Just like in the SDS page. But this time, we will be using an agarose gel. And next week, you will see the videos that I will be posting, no, on how this is done. Okay, so far, I think I've reached my goal for our video teleconference to explain exercise 7 and 8. So it's now your turn to ask questions and for clarifications. Questions and clarifications.
Sir, when will be the semifinal, sir?
Or semifinals, let's see. According to the calendar, if you can see this, it's going to be on the week of March 25 to 31. And I will be giving your group project in the next video teleconference.
Okay, sir. Thank you, sir.
Right, so I hope you understood the processes, no? Um, if I'm gonna ask you a question, I have a question. If you were the ones, if I'll ask you to perform those actually, will you be able to do them already? Because you watched the videos?
Depends po, sir. Kasi ma-shock ko, sir.
Yeah, that's the difficult challenge, no, with your batch right now doing Selmol because you only watch the videos. Okay, but you know the concept behind what we are doing, diba, is very interesting. In fact, a majority of the progress in the methodology in biology is already rooted with cell and molecular biology, no? So perhaps the department can find a schedule wherein you can really do the lab work yourself. Okay, I also cannot promise when that will be. But as of now, if you understood the process and if you know the purpose, then that will be enough for now. So, concerns, questions?
So, what sir, dili na gid mi maka-perform lugar sa experiment sir nga kami gid.
As of now, yes, you won't be able to. Because by the time face-to-face classes will resume, I think you will be taking the next subjects na. You will not be going back to CELMOL or any other subjects in the first sem. So, forward na talaga.
Yes sir, thank you sir.
With regards to your grades, I just ask for your patience and please wait patiently because I'm not yet done, really, with all of the grades, including the lectures even. So I'm doing my best to do them and at the same time prepare lessons. So I hope you understand that your midterm grade is going to be really, really, really late. But I assure you, I will do my best also to give justice to your work in the lab reports, etc. Yeah. Concerns, questions, clarifications.
Murag na ata, sir.
Right. So thank you for informing. And I thank you all for joining in this video teleconference. I mentioned this with the other groups already. It's very important to join video teleconferences. Not just because you're going to miss things if you don't. You will not because I will be posting this later on anyway. But why should someone attend a video teleconference? The answer is so that I can do the discussion. because I cannot do the discussion if there's no one, right? So, and there will be no recording. So I encourage you, and you tell your classmates also, to join video teleconferences, because it's really more fun if there are more of us in the video teleconference. So thank you very much, guys, for joining, and may you have a productive day ahead. Don't forget your schedules ahead. Thank you, and you are now all dismissed.
Thank you, kayo, sir.
Thank you,
Thank you,
sir.
sir.